I'd like to start by thanking Dr. Parmentola for inviting me to present some of our work. And I'm going to give you a bit of background on what our lab actually does in terms of basic science, and then try to inspire you with some ideas as to what this might lead to in terms of applications. You should know, just so you know me, I was trained as an engineer and a neuroscientist. And the problem that my lab has been interested in is this problem of visual object recognition. And so I like to start all my talks by showing you this movie here. If you watch this movie, you'll notice that as these frames come by, this is a rate of 6 frames per second. You can recognize one or more objects in each and every one of those frames, even though they're coming by at quite a rapid rate. This is really a remarkable ability we have to do this, something that machines do not yet have. Our ability to do this is very accurate, it's very fast, and it's very tolerant to variation. This is critical to the problem. This is why machines don't do this well and we do. So for instance, the illumination conditions on Yoda's head here. Now, what's really interesting about this problem is that it feels effortless to us. It's not like playing chess or doing long division. We don't have to be really, it seems, taught to do this. Our brains are essentially wired to solve this, and this means to us it's deeply important it's critical to our survival, and understanding it we think will give us deep secrets about how the brain works and how cortex works to compute things in general. And so we view it as a gateway problem to intelligence even. The practical mission of my lab is to try to contribute to an understanding of how the brain constructs an image representation, a pattern of neural activity, a code, if you like, that can underlie or support this task we call object and face recognition. How does the brain do that? I might want to step back first and tell you why we even do this problem. So you've heard today about cognition, decision-making, all the things we ultimately like to understand. But that's all resting on top of this currency of cognition we don't even understand yet. That is, the things that form, the things that get laid down in memory, as you just heard, the things we use to guide decisions, the thing we use to guide actions and make value judgments, those are object representations. They are not photographs in your head. That's not what's stored in your head. They're patterns of neural activity that stand for these objects. And we need to understand those patterns and how they're created. Understanding them will lead to understanding how we do many things, like these things shown here on the right. Many of these, of course, will have applications. Clinically, understanding how the brain solves this problem will, of course, lead to ultimately treatments for things like agnosias, which can result from things like traumatic brain injury, also have impact on Alzheimer's, autism, and dyslexia, we think. Again, I like to think of this problem as being a gateway to understanding canonical cortical computation, and from that point of view, there's a lot of really exciting technology applications, things that are obvious like machine vision and prosthetics, and I'll mention both of these, and also things that I think are less obvious to us at this point, but will become very powerful in the future. To step back on the problem of recognition, the reason this problem is hard for machines is that the same object can present an essentially infinite number of images to your retina. And this is due to variation in things like the object position, size, its pose, illumination, all of this natural variability. Somehow we deal with this, and somehow the brain can recognize objects amongst that natural variability. To do so, again, it must create a pattern of neural activity that still represents the object, still selective for one object among another, yet can tolerate or be invariant to this kind of natural variability. Our brains, again, do this effortlessly. Now, we'd love to understand this brain here, the human brain. We work on the other brain shown here, which is the rhesus monkey brain. This is not shown to scale. The monkey brain is much smaller than the human. The reason we like the rhesus monkey model is because of all the work of neuroscientists that have gone before us. We know where these interesting representations live. We know where the computations take place. They take place in a place called the ventral visual stream. It's shown here in green, and it projects down the temporal lobe of the brain, down to a place called infratemporal cortex, or IT cortex, you'll hear me call it. IT cortex is well positioned to project areas that you just heard about from Dr. Tien involved in things like memory, also to areas in the prefrontal part of the brain involved in decision and action. So we want to understand how these representations get produced in IT. So we know where they live in the primate. The other reason we like the monkey, beyond where we're having a roadmap, is that because these are non-human primates, we can study this representation and its precursor representations at the most appropriate level of abstraction. So that is, we can study the neuronal spiking patterns like you just saw in the previous talk. So briefly, what you should keep in mind is when an animal looks at a screen like this, if you present a visual object such as this car, then what will happen is you'll get a wave of neural activity that travels quickly along the ventral visual stream. And as I'll show you in this talk, you can quickly read out what that animal is actually seeing. And the interesting question to us is, what are the computations that allow that to happen? These are the kind of tools we use to solve or attack this problem, things like neurophysiology, behavioral studies in monkeys and humans, functional brain imaging, which you might call brain scanning in humans and monkeys, and overarching all of this is computation to analyze data and to build models of what might be going on. If you look at IT cortex and you go down and record single unit activity, so you put microelectrodes near neurons as shown here in infratemporal cortex, which is illustrated here, then what you'll find is that if you show, for instance, this series of 5 different images, and I could pick a bunch of random objects from a hat if you like, you'll see these little dots here. Those are called action potentials. You just saw those in the last talk. And you should just notice that there are more action potentials over here and less over here. That is, there's selectivity among this object set. If you look at other neurons, you'll find that they like other things, so they prefer other different images. So there's selectivity, and it's not all neurons like the same objects. You can also notice the time scale here. So this activity is arising about 100 milliseconds after the image hits the eye. So it takes the brain about 100 milliseconds to start putting the output of its computation up for us to see. And this is and it's complete in about 200 milliseconds. As I'll show you, we can read out the activity over these yellow windows that I've illustrated here. And this is all consistent with the movie I showed you at the beginning of the talk, your ability to do these kind of tasks very, very rapidly. So one way I can show this to you, and one thing we've done in a previous study, is to just take a large set of objects. These are 78 objects that I did with collaborators. And we presented those objects to the animal not just at standard positions and scales, but at a range of positions and scales to test this variability that I referred to. And basically, we can go in and record a whole lot of neurons, and you can plot the data like this way. And it's just to illustrate for you that we can collect a lot of data. The green intensity indicates the amount of response a particular site likes, and the rows indicate the 78 objects that I referred to. And you should say, OK, it's a big complex set of data. Is there anything interesting in there? Can we read out object identity? And this is very similar to what you heard from Dr. Chen in the last talk. The brief answer is yes, we can read out object identity very easily, very well. And I'll show you briefly how we do that. If you imagine this face being presented here, this would be the evoked pattern of neural activity that you would see across some n number of IT neurons, where n is a number we can vary in our analysis. We count spikes, build response vectors, a lot of detailed math you don't need to worry about. Basically, we ask how well can we classify this into different object categories. Here we had set up pre-assigned 8 different object categories. They were things like cats and dogs, human faces, toys, foods, and so forth. And so we build things called linear classifiers, which are basically machine learning techniques that take this high-dimensional data and try to find nice hyperplanes in that space to separate, for instance, faces from non-faces, to just read the code. And there's nothing magical about these tools. What's magic is that the neural data allow this process to work so well. So if you just look at the number of neurons you need to read out category in IT cortex, in about 100 neurons, you're reading out single trial accuracy that's closing on 100%. So this is single trial, cross-validated performance. This is what you could do reading all those neurons. You could read out the category of the identity of the object faster even than the animal could report it if that was something you wanted to do. Identification performance also follows quite closely. Moreover, this ability to do this, most importantly from the science point of view, tolerates things like the object's position and scale, again, the variation that we're so good at dealing with. It also tolerates other forms of variation that I won't have time to tell you about today. This is not possible in earlier visual areas. You can't do this from neural activity in the retina. You can't do it from neural activity in V1. And you can't do it as well in area V4. These are precursor areas to IT cortex. So we're looking at the output of a computation here. So if you've missed all of that, I'll just give it to you in one big summary here. What I've told you so far is object recognition is a hard problem for machines because there's this invariance. Objects produce many different images on the retina. And what I've shown you briefly in outline form is that the ventral visual stream, here shown in green, computes that solution, and that we can read a good, robust solution in IT cortex within about 150 milliseconds after that image hits your eye. So we can see an answer being sort of offered up here. We can sort of read that neural code. And the idea is that you have implicit object information on the retina. What's out there is sort of, unless you're in low light conditions or high occlusion, the object identity is apparent. It's apparent to all of us when I showed you those images. But it's somehow computed in a way that makes it explicit, that is easy to read for simple readout kind of tools that I just showed you. So that's the basic idea you should have from those kind of studies. But you say, well, okay, we know this now. What can we do with this kind of knowledge? Well, you could imagine if you want to go down the neural prosthetics road, you could say, well, we start putting in recording arrays. These are the kind of things we're using in the lab now. These are 100-channel arrays. You heard from Dr. Tien. You could put in lots of electrodes if you like. And then you could do nice things that might be interesting to you. You could say, well, if I wanted to read out the object faster than the animal could press a button, in theory, this data shows that you could do that if that was something of interest to you. You could, with harder techniques, but some of this has been shown for some classes of objects, reverse this process. You could say, suppose I want the animal to imagine this without showing it to him visually. In theory, with the right activation patterns, you should be able to write in object identity. And there's been, again, some examples of this. And I'm not going to dwell on neural prosthetics right now, but I'd be happy to talk about it in the discussion period. So given that you sort of see this code, those are things you can do. From a science point of view, what's most interesting to us, though, is understanding what's going on in all these arrows here, all these neural computations up to this solution as I described it. What's being computed and how is it being computed? That, to us, is the holy grail. I'll skip over what kinds of computations are going on. I'd just like to say that here's, again, the ventral stream laid out in horizontal form. Here are these brain areas again, now shown in colors. Again, a series of hierarchical processing through the visual stream. You've heard of that a moment ago. The way we think about this problem is you have millions of neurons in each of these areas, and each of them is like a re-representation, a new image of what's on the world. You have an image out in the front, and it gets converted to a new population pattern of neural activity. Perhaps with a canonical cortical transform along the way that I've indicated here by these letters T, and understanding that transform is to us, again, the holy grail of what's going on here. So we can do studies, and we are doing studies to analyze what's at the end of the stream, this IT representation that I talked about. But most interestingly, we're interested in what's happening step by step. What's happening as you go, for instance, from V1 to V2 to V4 onto IT. Again, with the idea that, hey, neuroanatomists have known for some time that these cortical areas, they all look the same. They might be carrying out a fundamental computation, and it's just iterated in a way that produces a nice form of representation. Now, to sort of put more flesh on that idea, we've been exploring ideas of how this transformation might be constructed, not just sort of saying, well, there's something here. Let's think about what could be going on. What are the computations? And so I'll give you an example of a study we've been doing in that regard, or one we just completed that's opened a new exciting line of research for us in this regard. And this is the idea, you've heard that the brain is plastic. But so one of the ways the brain may learn to do this object recognition problem, to solve this variability, is that it can see these objects. It can use the natural temporal statistics of the world to learn about what an object is and what its variability looks like. So this is a movie clip from Law & Order detective show. This is Detective Briscoe, as you probably know. Notice he's there for the whole several seconds of this clip. But if you cut out frames here, you'll see that in nearby instances of time, what's on your retina, which is essentially what this image is, you can see that Detective Briscoe's head is sort of translating across your retina there. So, and here you can see it also for size and also for pose. So again, identity-preserving transformations happening over short time scales where object stability exists over long time scales. So leveraging those temporal statistics and learning about what is invariant, what's an object, what's invariance, has been an idea that's been in the computational literature for about 15 years now, but hasn't been looked at neurally and hasn't been implemented in a working high-scale system yet. So we've been pursuing this idea because it seems very powerful, and what we've found so far is that your brain seems to be doing something like this. And I'll briefly outline for you what we do here. We found this result by manipulating the visual statistics during saccadic viewing. So a lot of your visual experience when you look around the world at objects is something like this. You probably don't realize this, but you're scanning the room, you're scanning the slide, you're moving your eyes about 3 times per second. You don't even realize it, but it might look something like this. So when you undergo this scan path, these rapid eye movements, what saccades do is they, again, cause translation of an object or its features over short time scales. Again, just as I showed you on the last slide. Now, we can, in the lab, reduce this to simplified conditions, say no background. And then the trick we play is both in humans and in monkeys, we play statistical tricks, which during the saccade, we have real-time eye tracking. We know exactly where they're looking. And we'll do things like flip the object mid-saccade like this. And in humans, we can do this in a way that don't even notice that we're doing this. We're basically playing statistical tricks under the radar, if you like. Now, so what we found from this, and I'm not going to show you the data, is that these kind of manipulations cause predictable changes in human object perception, the kind of changes that if they're using this kind of learning are the kind of changes we would have predicted. Our lab has shown this along with other labs, and we've recently found that it can change the IT neuronal properties that underlie that recognition code I showed you earlier, what's called their invariance or their tolerance properties. And we recently found this result, and we can track these changes of neurons while we're manipulating the statistics. What's really exciting about this from a machine point of view is that this learning is completely unsupervised. We didn't have to tell the animal to learn. We just expose him to the statistics. And that's powerful because these kind of statistics that you can learn about objects in the world, you're getting lots and lots of this, about 100 million such saccadic learning events each year. So that's a lot of data to sort of leverage and to build up these good kind of representations. And we think the kind of things that Cortex might have evolved itself to take advantage of. So what I've told you is that we can read a good representation in IT Cortex. And what we'd like to do going forward is say, OK, how does a visual system build this? This is the neuroscience question. This connects in a reverse engineering mode to the computational scientist question, which is how do you build machines that can do this thing? And we'd call that reverse engineering. These form hypotheses that can be used to guide our experiments. And forward engineering take these models, if you like, to real-world applications, things like artificial vision. So that's one of the exciting roads that we're connecting with now. I'm going to just show you a little bit about what's going on in the modeling direction there of sort of trying to implement these things in machines, these bio-inspired algorithms. And this is an example from neural data all the way back from 1960s. Hubel and Wiesel sort of laid the groundwork for these kind of algorithms. And they're basically stacked operations where you have a series of and or-like operations cleverly arranged that build up selectivity for object identity and tolerance or invariance to those kinds of transformations that you must deal with. Now, again, I won't take you through the details, but a number of labs have implemented these in more computationally oriented frameworks. And most notably recently, my colleague Tommy Poggio at MIT, his group has implemented a model of the ventral visual stream that captures a lot of these ideas, iterated hierarchical processing and or-like operations. And I'll blow that model up for you here. And what's cool about this approach is that even this sort of first pass rudimentary form of this algorithm can match a lot of the IT data that I showed you earlier in the talk. It can also do things like more complex tasks like animal, no animal tasks here. So is there an animal? And here are distractors. So these are real world kind of images. And they had a range of different kinds of animals that they used and so forth. Again, I won't take you through the details, but if you line that up against human performance, at least under very limited human viewing conditions, which is an important caveat, but under limited human conditions, these kind of bio-inspired algorithms show performance that's similar to human performance. So they say, well, maybe this is on the right track. Tommy Poggio's group has extended this towards scenes. This might interest folks interested in things like pedestrian and vehicle detection and finding things like buildings, things you might want to put in an autonomous vehicle. And it does not perfect, but reasonably well. We have to still keep digging. We're still missing a secret ingredient. Someone mentioned the physics work earlier, suggesting there's something magical about neurons that we just haven't discovered, or something, whatever you like. You can dream. I can list a bunch of things here. So how are we going to get out of this idea? Well, we can keep doing experiments, looking for some hoped-for thing, or we could sort of take a practical approach and say, well, in theory, this kind of thing is testable. If we take these ideas and implement them, well, we can say, does it get us human-level performance, or does it not? But in practice, what I just said is really, really hard to do. And that's hard for sort of two basic reasons. One is that we first don't have good benchmarks of what a good system should or should not be able to do. And so even just defining benchmarks of what the task is turned out to be quite challenging. And that's something we've been working on and we think we have good progress on in terms of focusing on the invariance problem. I won't dwell on that today, but I'll sort of tell you more about the second half of the problem, which is if you want to test all those things, as sort of Dr. Marder mentioned in her talk, there's lots and lots of parameters, high dimensional space that we're working in here, visual input, lots amount of visual experience if you believe what I told you about that. And when you say principles, as I outlined a moment ago, that doesn't give you an engineering spec that says, well, you need this many synapses here with those and those weights. You need to search that space or try different things. So here's our class of models. It's a very, very large parameter space, and it's still just a subclass of all the potential models you could build of a sort of hierarchical system that's meant to model parts of the ventral visual stream. Again, lots of parameters to explore. I won't take you through those. But what's very cool, and this relates to Dr. Kurzweil's talk, if you saw yesterday, is that the CPU power, and most impressively, the GPU power of machines is growing tremendously fast right now. And it's on a point where now we can have in our lab, we sort of are almost getting 300 to 1,000-fold speed up over standard MATLAB implementations, which is how vision researchers have been modeling these things to date, using this kind of technology. So we're getting a lot of computational speed up to test these kind of models, and that's kind of potentially game-changing for us. I mean, here's CPUs. GPUs are, again, accelerating dramatically here in terms of what they can do when they're doing things like visual processing, which is the same kind of operations iterated over lots of space. So this kind of high-performance parallel computing is opening for us new doors and exploring models for us. So testing models which begin to approach the scale of natural systems. We can test many thousands instantiations of these models. We think millions in the next few years. And we can screen off the best performing models and then ask what's working well about them. And here's an example of how we've been doing that so far. And we're getting sort of reproducible performance when we screen this way, much like high-throughput biology screens. So this is a tool that computation is opening up. We can use this power to also lean on the experience side of things and, again, provide lots of visual experience. And in theory, we think we can discover powerful new forms of visual representation for applications that we're already thinking about, and perhaps, just to speculate, maybe things about neocortical computation for applications, again, we can only dimly imagine. So if all that went by too quickly, all you need to do, or remember from my talk, I'm going to put on this slide for you here today. So we know where the brain computes the solution to visual object recognition and face recognition. To a first order, I think we can say, and some of my neuroscience colleagues will disagree with me, but I think we understand the basic nature of that neuronal code, and I'd be happy to talk about that in questions. This, in theory, could allow neural prosthetics. Again, I'd be happy to talk about this as to what the pros and cons are and what the limitations are over there. We know also many principles of how the solution might be computed, motifs of visual processing, things that are important, things that are not important, things I briefly mentioned. Even immature implementations of those seem to have surprising power when they're actually instantiated in systems, and I mentioned this with Dr. Poggio's work. So this starts to get towards artificial vision, and what's cool is that we build systems like this. Unlike people, they never get tired, even if they're doing boring tasks. And so what's really exciting now for us is rapidly rising computational resources are opening new doors and evaluating these kind of principles. And this might lead, we think, to new discoveries. So right now, with these rising computational power, the potential for discovery is very high, and both for applied artificial vision, we think at the moment, and if I just speculate in the long run, if we think about cortical computation and what it means to be intelligent, what it means to be human, that's the thing we dream about. We're not working on it at the moment, but I'm going to throw that out there as something that we think this work will ultimately connect to. I'd like to thank the folks whose work I mentioned here today, especially Tommy Poggio, shown here, colleague at MIT. And these are all folks in my lab that contributed to the studies I showed you here today, and also my funding agencies. And thank all of you for your time. Thank you.